All right, let's jump right in. Welcome to this explainer, where we're going to treat college-level cell biology like a high-stakes microscopic detective story. If you've ever wondered how scientists actually figure out what the heck is going on inside a living creature, well, it all comes down to the toolkit they use to crack the case. Today, we're exploring the absolutely incredible machinery of eukaryotic cells. We're going to look at how we observe them, how we literally pull them apart, and what we find when we do.
To keep things structured, here is our roadmap. We'll start with the unseen microscopic world, move on to the biologist's essential toolkit, check out a diversity of shapes, dive deep into the fluid mosaic boundary, and then wrap it all up with a quick summary.
Okay, Section 1, the unseen microscopic world. So we have to start our investigation by stepping back in time a bit, because our knowledge of life is entirely tethered to the technology we can build. We can only really understand what we can observe, right? It actually took over a century of technological refinement to realize that every living thing, plants, animals, us, is built on the exact same foundational unit. Think about it. In 1667, Robert Hooke investigates a tiny piece of cork, sees this honeycomb structure, and coins the word cell. But then it took until 1838 for Schleiden and Schwann to formally establish that both plants and animals are made of these cells. And finally, in 1858, Virchow cements the idea that all cells have to come from pre-existing living cells. And you have to appreciate the sheer, unbelievable dedication required to see this hidden world back then. Antony van Leeuwenhoek, often called the father of microscopy, he didn't just go out and buy a microscope. No way. He made over 500 optical lenses and built at least 25 single-lens microscopes completely by hand. Through these tiny brass instruments, he became the very first human to witness an entirely hidden, bustling world of bacteria and red blood cells.
Moving on to Section 2, the biologist's essential toolkit. Fast-forwarding a bit, how do modern biologists investigate the inside of these tiny living structures today? Well, looking isn't enough anymore. We actually have to take them apart. I want you to think of the eukaryotic cell not as some sort of watery balloon, but as a dense, incredibly high-tech manufacturing plant. There are distinct departments, organelles, and each one performs highly specific tasks, from generating energy to packaging up proteins. But to truly understand the chemistry of this factory, scientists need a reliable way to physically bust it open and isolate the different machines. Enter subcellular fractionation. Specifically, a really cool technique called differential centrifugation. This is a massive, massive leap from Leeuwenhoek's tiny glass lenses. Essentially, this method acts like a highly calculated, ultra-high-powered blender. First, we break the cells into a suspension, basically a cellular soup. Then, we spin them. By spinning this mixture, we use intense gravitational force to pull the heaviest factory components right to the bottom. The really clever part here is the system behind it. By spinning this soup at progressively higher and higher g-forces, we effectively sort the factory entirely by size and density. A spin at 800 times gravity, that pulls down the heavy, dense nuclei. Then we take the leftover liquid, the supernatant, and spin it way faster at 15,000 times gravity to isolate the mitochondria and lysosomes. We can even ramp this up to a staggering 100,000 or even 200,000 times gravity to isolate tiny membrane fragments and ribosomes. This is absolutely essential for modern biology because it lets us test the specific chemical functions of isolated organelles without the rest of the cell getting in the way.
All right, Section 3, a diversity of shapes. So, once we use these incredible microscopes and centrifuges, what kinds of cells do we actually find out there? When you first learn biology, it's super easy to picture all cells as these perfect little spheres or neat cubes. But the reality is that form heavily follows function. Nature absolutely does not build one size fits all. It engineers highly specific architectures for highly specific jobs. Depending on their exact job in the body, cells take on some wildly different shapes. Take our human erythrocytes, our red blood cells. They're these biconcave discs, which is genius because it maximizes their surface area for carrying oxygen and lets them bend and squeeze through tiny microscopic capillaries. Fibroblasts and muscle cells, they're spindle-shaped so they can stretch and contract. Your skin relies on flat, squamous epithelial cells for layered protection. Meanwhile, your brain relies on multipolar neurons, which have these incredibly long branching structures, specifically designed to conduct electrical signals across vast distances.
It's not just the shape. The scale of these things is staggering. Just look at the size differences nature has cooked up. On one end of the spectrum, we have mycoplasma bacteria, which are basically pushing the absolute microscopic limit at just 0.1 to 0.3 micrometers. Then, on the complete other extreme, you have the aptly named Chaos carolinensis. It's a giant amoeba that measures up to a massive 5 millimeters. Think about that. That is a single-celled organism so colossal, you can basically see it with your naked eye.
Let's move into Section 4, the fluid mosaic boundary. So we've looked at the cell as a whole, but now let's zoom all the way into the very edge to explore the barrier that protects our microscopic factory. This is what dictates exactly what gets to go in and what has to stay out. For a really long time, scientists weren't quite sure how to visualize this selectively permeable border. That all changed in 1972 when Singer and Nicholson proposed the fluid mosaic model. It was a total game changer. Instead of a rigid wall, actually scratch that, it's nothing like a wall, they accurately described the cell membrane as a highly dynamic, two-dimensional fluid. If we break down the structure of this mosaic empirically, it's just this beautifully balanced mix. The plasma membrane is roughly 45 to 50% lipids and equal 45 to 50% proteins, and then sprinkled in is a smaller amount of carbohydrates, sitting around 4 to 8%. It's a highly engineered ratio designed to perfectly balance physical protection with cellular communication. The entire foundation of this boundary relies on the most common lipids present, phospholipids, and they are fascinating because of their dual nature. They have hydrophilic, water-loving heads and hydrophobic, water-fearing tails. Because both the inside and the outside of the cell are essentially watery environments, these molecules naturally, automatically arrange themselves into a protective double layer, safely hiding their water-fearing tails right in the middle. But remember that word fluid in the fluid mosaic model? Like I said, this is not a static brick wall. The dynamic movement of these lipids is wild. They are constantly shifting laterally, just swapping places with their neighbors. They rotate in place. And occasionally, they even undergo what's called a transverse flip-flop, where a lipid jumps entirely from the inner layer over to the outer layer. It's this continuous, restless movement that gives the membrane its vital flexibility. When you put the whole shebang together, you get this magnificent, living structure. Embedded within that shifting sea of phospholipids, we've got cholesterol molecules acting as a temperature buffer, so it doesn't freeze solid when it's cold or melt apart when it's hot. And floating amongst all of this are massive integral and peripheral proteins acting as physical transport doors, receptors, and communication hubs. It is an incredibly sophisticated boundary that is reacting to its environment in real time. Finally, Section 5, Summary and Next Steps. We've covered a tremendous amount of ground in our biological investigation today, so let's piece this macroscopic and microscopic puzzle together. What should you take away from this explainer? Well, centuries of observation using increasingly advanced microscopes allowed us to uncover an incredible diversity of cellular shapes and sizes. Then, modern separation tools like differential centrifugation gave us the critical ability to isolate and study the exact jobs of those internal organelles. And lastly, all of this complex machinery is contained, protected, and regulated by the wonderfully dynamic lipid-protein barrier we call the fluid mosaic membrane. I want to leave you with one final provocative thought. Think about Leeuwenhoek. He had a tiny piece of polished glass, and he discovered cells. Today, we have massive electron microscopes and ultracentrifuges revealing a bustling molecular city. So as our technological toolkit continues to evolve over the coming decades, what entirely new mysteries of the microcosmos are just waiting to be uncovered right inside of you? Thanks so much for joining me on this explainer, and keep questioning the unseen world around you.